Welcome back. This is Dr. Blinch from 2025, and today we're going to look at a continuation of blind sight, which was the loss of your cortical visual pathway, and we saw the distinction between the cortical and the subcortical roots. Today what we're going to do is we're going to look at, well, what happens if you lose half of your cortical visual pathway? And we'll look at one half versus the other half, and those are known as the dorsal and ventral visual streams. So just to review here, we have our cortical visual pathway, the solid lines, that go to the primary visual cortex, and we hadn't really made it past there, but visual processing continues on into many areas of the brain, and that's what we'll look at today. The subcortical root, previously in the dashed lines here, had stopped at the pulvinar nucleus in the brainstem, but it also continues on into the cortex, and we'll see that as well. So, simplified diagram here, reviewing the conscious perception from our cortical visual pathway, and then unconscious perception for action via the subcortical pathway. But just like I mentioned in the past figure, processing continues on after the pulvinar nucleus, and there's a ton that happens after the primary visual cortex. That's what we're going to investigate today. So here's a basic diagram. So imagine this, around here we have the primary visual cortex, and visual processing continues largely in two areas. One up into the parietal lobe, and we call this the dorsal stream, and also down here into the temporal lobe, and we call this the ventral stream. And we're going to see what is the separational functions of these two streams. When do we use one? When do we use the other? And also how do we use them typically simultaneously. Now, why is this pathway the dorsal stream? It might seem a little weird from human anatomy, but if you think of yourself like a fish, or think of a fish, and here is thinking of yourself like a fish. So to be a fish, your head's got to be tilted back like this, right? Humans have evolved to be standing upright with their heads forward, whereas if you think of a monkey or a chimpanzee, they're down on all fours and their head is rotated back. So if we go way back in evolution to being a fish, it makes sense that a fish, the dorsal fin is on the back. Now, if you continue up the back and you get to your brain, so now we're at the kind of the top of our head, but that's why we call this the dorsal part of the brain, because in other animals that would be rotated back and it would be kind of in line with your dorsal fin. So hopefully that helps you to remember, dorsal is this part of the brain, so then the lower part of the brain is going to be ventral. So here's the basic schematic of the visual processing that occurs after the primary visual cortex. So we have the dorsal stream, and there's processing all along here, and eventually that terminates in the posterior parietal cortex. So we're in the parietal cortex, and we're in the more posterior aspect of it. Then we also have our ventral stream, and there's processing all along this, but it terminates in the inferotemporal cortex. So if we go to our temporal cortex, we're talking about the more inferior or kind of lower parts here. One interesting thing is, if you note, there's two ways to get information to the posterior parietal cortex. There's via the primary visual cortex and the dorsal stream, but there's also the subcortical route. There's kind of this redundant pathway that allows information to get to the posterior parietal cortex. And that was used in blind sight. So some blind sight abilities are driven by visual processing in, say, the superior colliculus, the pulvinar, but even with the primary visual cortex damaged, and no information flowing, say, down the traditional dorsal stream, there is information that reaches the posterior parietal cortex via the subcortical pathway. Now there's an interesting distinction between these two streams, and the brain seems to divide and conquer. It says, okay, dorsal stream, you're going to do this, ventral stream, you do a completely different function, and typically you're both operating at the same time, and together you'll do all the visual processing. So the dorsal stream is our action stream. It is what allows us to use vision for action. And we'll see lots of examples of this. Now we sometimes call this our how abilities. So an example could be, how am I going to use vision to guide my hand to my mug? Well, the dorsal stream will be processing the visual information and relaying that to my motor centers so I can use vision to guide my action to pick up that mug. So you can think of this as visually guided action, which we use a lot throughout the day. Now, the the ventral stream is for perception. So this is the information that goes to the inferotemporal cortex. And we also call this our what abilities. So we're using vision and we're trying to answer, what is that object I see? And here's a funny example from my desk. This is a Christmas elf door hanger that happens still to be here from months ago. And if you see this, you want to know, what am I looking at? Oh, I need to identify it's an elf. Maybe I need to identify that it's made out of foam. Those are all being mediated by information that goes to the ventral stream, our what stream ending in the inferotemporal cortex. We're going to look at two ways to understand the dissociations of these streams. And the first is by looking at two unique individuals. One, the first, DF, has damage to their ventral stream, so their dorsal stream's completely intact. And the second, RV, will have the opposite condition. We'll save her for after we cover DF. So DF, her condition is called visual form agnosia. So she's going to have no problems using vision to guide action, but when she looks at an object, she can see it. So she has a lot of these, lower visual areas like the primary visual cortex. So she's not blind, she can see things, but then putting that information together and saying, well, what is it that I see? That part is damaged. So visual form agnosia. Agnosia means without knowledge. So the idea is she can see things, she can see their visual form without knowledge of what they are. So DF was a middle-aged woman at the time of an accident. She had suffered Agnosia, or lack of oxygen to the brain from carbon monoxide poisoning. And thankfully it wasn't fatal, but it permanently damaged her ventral stream. So she's got visual form agnosia, as I mentioned before, and we're going to see that she can still see things like color and texture, but then saying, well, what is it that she sees? What do those individual colors and textures make up? That has been lost from permanent damage to her ventral stream. Let's look at an example. Oh, sorry, one thing first. Just to be very specific about DF, a little summary slide here. Saying here, ventral streams permanently damaged. So she's going to be challenged with these what tasks. Dorsal stream completely intact, so she'd be normal as any other neurologically intact individual at using vision to guide her action. So preserve visual motor control, your how abilities. All right, let's get into some examples. So imagine we show DF objects, and the first example is this flashlight. We show it to her and we say, hey, do you see this? And unlike someone with blind sight, she would say, yes, I can see that, I'm not blind. And she can see the individual aspects of this. So this is a quote from her. She said, it's made out of metal. Yeah, it is made out of metal. Is it aluminum? Yeah, I think it is too. It's got something red on it, looks like plastic. Okay, so she can see color. She sees this shiny texture. She can see that this has a plastic texture to it. But then for you or I, the ventral stream would then immediately tell us what this is. It's a flashlight. This is easy. But for her, she doesn't have that ability, that kind of highest order visual ability to put together these individual features into this cohesive whole, or to answer this what question. So she just guesses. And she's in here, she says, is it some sort of kitchen utensil? And that's not a bad guess, right? Something metal with plastic on it could be a kitchen utensil. And if we show her something, an object, say, in a kitchen, she's going to have a better likelihood of guessing using contextual cues. So if we want to make it really hard for her, we have to, bring her into a plain room, a testing room, so say in a doctor's office, and show her all sorts of random objects from not just the doctor's office, but from you might encounter in everyday life. And then it's going to be very hard for her to identify what these things are, because it would lack any sort of contextual cue. Like, say, if you're being in a kitchen and you pick up something metal and plastic, well, it's probably a kitchen utensil. This quick video about not DF, but another individual with a type of object agnosia, very similar to visual form agnosia like DF. And what's happening here is this is the individual kind of testing the person with object agnosia. And to make it really hard to guess, instead of having the physical object on this flip book, they just have these like pictures, these 2D cartoon images of objects. Without the 3D information, the color information, it makes it even harder to kind of guess what the individual is looking at. And in this case, they're showing them a combination lock. The video kind of picks up midway, and earlier on in the video, the individual was convinced it was a telephone. So the tester said, okay, it's not a telephone, and because you're really struggling, I'm going to give you a list of 3 options. You know, is it A, B, or C? And that's where the video will start. So take a look how he struggles to identify what it is because he's damaged his ventral stream. But note his hands seem to know how to interact with it. And that would be his intact dorsal stream that eventually gives him the information about what he's looking at despite having a damaged ventral stream. So check out that video. The link is in the description. Here's an example of a few more tests with DF. And this is like the flip book we previously saw in the video where we show you these 2D drawings, and it's very hard to guess what they are without the 3D hints and the color information that could help lead you to the solution. So we show DF a picture of an apple. We say, what is it? I don't know. Okay. Can you copy this picture? Now, she can use her hand to draw. So it's not that she's lacking in her visual motor abilities to draw because her dorsal stream's intact. The problem is she doesn't know what she's looking at. And if you don't know what you're looking at, then it makes it very difficult to copy it. Then the question is, well, has DF forgotten what an apple is? We say, okay, do you know what an apple is? And she says, yeah, of course, before my injury, I've seen lots of apples. I know what they are. Okay, great. Can you draw us a picture of an apple from memory? And yes, here we see that her visual motor control is intact. So her dorsal stream allows her to make a drawing. So it's not that she couldn't draw, it's that she didn't know what she was trying to copy with a damaged ventral stream. And we get similar results if we show her a book. She knows what a book is. She can draw us a picture of a book, but she can't tell what she's looking at. So when DF presented to the doctors, they were kind of at first looking at everything that she had lost. Showing her things, she didn't know what they were, but she could remember these things. It was kind of fascinating to the doctors, this deficit. And then what they realized during these tests is that, and they didn't realize this at first, is that her visual motor abilities, her dorsal stream was completely intact. And at one point they had a pencil and they said, okay, what is this? And kind of like the flashlight, she'd be able to say, oh, it's yellow, looks kind of like wood. There's a pointy tip, some metal on the end, and maybe from that she could guess that it's a pencil. But without a ventral stream, she doesn't get that kind of snap judgment of, oh, that's a pencil. That's easy for you and I. We don't have to look at this and struggle to think what it is. So like, okay, she can't recognize this object. They said, okay, pick it up. And they thought because she couldn't recognize it that she would pick it up with a very fumbly grasp. But she made a very coordinated grasp. If they held it like this, she would pick it up appropriately like that. And if it were this way, she would come in and adjust her grasp to pick up the object. And that's what we expect from a neurologically intact individual. So they were shocked. How could she not know what this object is, and yet her hand can go pick it up just fine? And that's when they realize that there's this dissociation between visual functions between the ventral and dorsal streams, and one can be damaged and not the other. So this led to a series of experiments by Milner and Goodale. They're both famous for showing the results from DF and later RV, and showing this distinction between these two visual pathways. So they developed a lot of experiments. We'll look at just a few, where they're going to ask you to do one task with kind of two different types of instructions. So in this case, we have what we kind of call the mailbox task. So imagine this is like a mailbox in front of you. It has an opening, and you have a letter here, and your job is to, either show us, match the orientation of the mailbox with the letter. So if the mailbox is here, and if this is your letter, you should rotate the letter to be the same orientation as the mailbox. And if you're neurologically intact, if you have a ventral stream, this is very easy, right? If the mailbox is here, I can easily say, well, that's the mailbox. Now the second version of the mailbox task, now instead of trying to match that orientation, we just say, go ahead and mail the letter. The interesting thing about this is knowing the orientation of the mailbox. That is a what ability, knowing what is the orientation of the mailbox. So it's a what ability, and it's driven by the ventral stream. To then actually use your hand, to use visual feedback to guide your hand to the mailbox, that is done by the dorsal stream. That's one of these how tasks. So in URI, if both of your dorsal and ventral streams are intact, you can do both of these very easy. But for DF, she has an intact dorsal stream, so she'll be able to mail the letter just fine. But remember, her ventral stream is damaged, so she's not going to be able to tell you the orientation of that mailbox. And it's kind of shocking because, well, if you don't know the orientation of the mailbox, how can you mail the letter? And that's because, well, mailing the letter requires one part of her brain that is intact and can do that, but that part of the brain, the dorsal stream, can't then tell the broken ventral stream, hey, by the way, this is the orientation of the mailbox. It's almost like you've got two people in your brain, one in charge of one type of visual ability, one in charge of the other, and they don't really communicate with each other. Or I guess in this case, one person is missing, damaged, and you only have one person remaining, and they can't take over the functions of that person who's gone. So in this task, imagine this is the orientation of the mail slot. So here, a perfect response would be that it's vertical, or mailing it would be, mailing it, like this. Now, if DF posts the letter like this, you can see this is obviously poor performance. They're not very close to vertical at all. Now, in the experiment, every trial, the mailbox had a different orientation, so that if it was always the same, it would be easy. So on this trial, the orientation is horizontal. They did, imagine any of the 360 degrees. And here, let's say, you're again way off. So it's supposed to be horizontal and you're vertical. To compare all these trials to each other, what we're going to do is we're going to line them all up as if all the trials were vertical, even though every trial had a random orientation. So let's just rotate this one to be vertical. But imagine we had, a whole host of trials here. We're going to overlap them, and what you can see is that, well, from our two trials, this is poor performance, because they should be vertical, as indicated by this triangle, and they're not. So this is an example of two trials that have poor performance. It's just showing you that to kind of orient you to how these figures work here. So before we get to DF, let's look at age-matched controls, neurologically intact individuals. And again, these tasks are easy. So if we ask someone with a ventral stream and a dorsal stream to tell us the orientation of the mail slot, then they get this right every trial. It's easy. And then if we ask them to go ahead and post the letter on a separate block of trials, they can also do that. These tasks are easy. But let's look at DF. So DF, ventral stream is damaged. Now to do the what task, to tell us the orientation of that mail slot, she needs her ventral stream. It's damaged, and so she's horrible. And if we ask her on any trial, we say, okay, where do you think the mailbox is? She might say, here. And you know that's not right. We say, how confident are you? She's like, I don't know. So she knows that she's not good at this task. She's basically just guessing. She's all over the place. If we look at her posting though, so we say, okay, don't tell us the orientation, just go ahead and mail it. She will accurately guide her hand and the letter into that mail slot, just almost as well as control, a little bit worse, but way better than this, horrible guessing we saw when she was trying to do perceptual matching with her ventral stream. So we got a huge dissociation between performance here. She's perfectly good at posting because she has a dorsal stream. When she tries to match the orientation without actually posting, she's horrible. She just guesses. Another example with DF, and here we're going to rely on the fact that when we reach for an object, we scale the size of our grip aperture, so that like the distance between my thumb and finger, to the size of the object. So if I'm going to pick up this pencil, I'm going to make a small grip aperture because it's a small object. And I don't even really think about that. That's just kind of naturally how grasping works. And then if I have to pick up something much bigger, well, I'd probably make a different type of grasp, but just for argument's sake, if I were to just put my thumb and index finger on it, then I'm going to have a much bigger grasp to pick up that object. So over and over, lots of studies have shown that we scale the size of our grip aperture to the size of the object. And we're going to look at that in DF. This is an example of a control neurologically intact individual, and we give them 3 different size objects. So you can think of this as a pencil. This may be, let's say, a paper towel roll. Oh, my dimensions might be off here. And then 5 centimeters. Well, we gotta go bigger than a paper towel roll. Let's go with our water bottle. Okay, definitely not 2, 3, and 5 centimeters, but just think about a small object, a medium object, and a large object. And we can actually track our grip aperture over time, which is what we're seeing down here on the x-axis. So the participants probably start with their fingers kind of together like this, and what you see as you go to pick up an object is your grip aperture increases in size. It gets to a maximum size, shown here, and then as we get closer, it kind of narrows in down to the size of the object. And we can analyze this whole path or this whole trajectory, but to keep it easy, what we're going to look at is just the size of the maximum grip aperture. And although this is larger than the object itself, it will scale to the size of the object. If I give you a small object, you make a small maximum grip aperture. If I give you a large object, you make a much larger maximum grip aperture. And that's what we'll look at here with DF. So we're going to need 2 versions of this task again. One where you're using your ventral stream. And here, so imagine it's kind of like a domino in front of you. And we just say, estimate the size of that domino. And on some trials, the domino would be bigger and some would be smaller. So DF she will try to estimate the size of the object. We'll see she's bad at that because her ventral stream is damaged. Then we'll ask DF to grasp the object. So don't estimate the size, just go ahead and grasp it. That requires the dorsal stream, and she'll be good at this task. Here's a control individual, so someone with a functioning dorsal and ventral stream. So let's look at grasping first. Remember, this uses the dorsal stream, and we have a small domino and a large domino. So just 2 versions. And what we see is that if we give you a small object, you make a small maximum grip aperture. If we give you a large object, you make a large grip aperture. That's good. You're scaling the size of your grip aperture to these 2 objects. Instead of grasping it, we just say, well, how big do you think it is? How big is the small one? How big is the big one? And again, we're going to see that you'll scale the size of your grip aperture to the size of the object, but this you would do with your ventral stream. Here's DF. So she has a dorsal stream, so when we ask her to actually go ahead and pick up the object, she's good. She scales the size of her grip aperture, just like our control participant. But now we say, okay, how big is that object in front of you? And you would think because she can pick it up that she'd be able to answer that question, but to do that she needs her ventral stream, and that's been permanently damaged. And what does she do? She kind of opens her hand big on every trial. And if we had video of this, you would see that her reaching, unlike a normal grasp that looks very smooth and coordinated, would be very fumbly. She probably opens her hand big right off the get-go and just kind of hopes to touch the object. Because if she makes her hand big, it's more likely to touch the object. And if she can touch it, her sense of touch is intact. You know, she's damaged a visual area, not touch. Touch is fine. So if she can touch it, she can then use her intact sense of touch to grasp that object. Just like if I were reaching something with my eyes closed, you know, if I can get my hand there, well, once I can feel it, I can then grasp it using my sense of touch, even with my eyes closed. So these little cylinders here are supposed to be her thumb and index finger. And the idea is when we give her a small object and we say grasp it, she makes a small maximum grip aperture, which is good. And then when we give her a bigger object, she makes a bigger grip aperture. So she's scaling the size of her grip aperture to the objects when grasping, which is using her dorsal stream. Now, when we ask her to do a perceptual estimate, hey, how big is that small object? How big is that large object? Remember the results in both cases, she just opens her hand big. So this is poor scaling. She is not scaling her grip aperture to the object, and that's because her ventral stream is damaged. So quick summary of DF here. She has visual form agnosia. She struggles to know what an object is. She's not blind. She can see individual aspects of the object. But all that information coming together to tell you, oh, I'm looking at, say, my Christmas gnome, you know, that is lost. And despite that loss, using vision to guide her actions, so she then wants to go up and pick up the garden gnome, the Christmas gnome, Christmas elf. Oh, I need better visual aids here. She can pick it up just fine because that's a different part of visual processing which is completely intact. That is our first individual. We're going to look at the second individual, and her name or her initials are RV, and her condition is optic ataxia. She is quite literally the opposite of DF. So RV, her dorsal stream is damaged. So all those visual motor abilities that DF had, well, RV is going to struggle to do those tasks. And the things that DF struggled to do, identify what she was looking at or make perceptual estimates of size and orientation, RV is going to be completely fine at these because her ventral stream is preserved. Now RV, just by coincidence, was also a middle-aged woman when she had brain damage. So DF, RV, both middle-aged women, that doesn't really help to distinguish between them. The difference with RV is that it wasn't carbon monoxide poisoning. She had a couple of strokes, and these resulted in permanent damage to her dorsal stream. And these are, MRI images showing that damage, but I can't really point out on here easily where that actual damage is. So it's like, okay, well that doesn't look good, but if that's damaged, then this really doesn't look good. So if you end up becoming an expert at this as you continue on your career, send me an email and let me know, can you see the damage in here? I'm not sure. Just a summary slide about RV, just to be very clear about her condition. It's referred to as optic ataxia. So ataxia means poor coordination, and her coordination was fine. The problem is when she tries to use vision to coordinate her movements. So it's ataxia, poor coordination, but they add the optic part because it's poor coordination using vision or poor visual motor abilities. So you can see here the damage here to her dorsal stream. Ventral stream is intact. So we ask her what questions, what do you see? She'll have no problem. So let's look at RV on many of the same tasks we looked at with DF. So here's an example of her poor visual motor control. So let's say we ask a participant to pick up a golf ball. This is a control individual. And what you'll notice is that the hands are very coordinated. So not only do we scale our grip aperture, but if we're picking up something like a golf ball, all the fingers are going to move in a coordinated way to pick that up comfortably and efficiently. RV does not have that because her visual motor control via her dorsal stream is damaged. So here, notice that she's just kind of tries to make her hand large and really hopes to touch it. So similar to what we saw with DF. Now in this condition, she makes her hand large and her visual motor coordination is so poor that she misses the object entirely. So if you just look at how she controls her movements, it's very obvious that she has poor visual motor control. Let's look at the same task we gave DF. So we show her a picture of an apple and we say, what is this? Because it's an apple. So to do that, you need your ventral stream and her ventral stream's intact. Say, okay, what do you see here? Oh, that's a book. Great. What's this? That's a boat. Okay, great. No problem knowing what an object is, even when it's difficult, without 3D clues and colors to help you identify the object. Okay, so she obviously knows what an apple is. Can you draw us a picture of an apple or a book or a boat? And these pictures aren't great. Maybe they'd be okay for a 3-year-old, but the reason she can't draw them very well is because she struggles to use vision to guide her actions. So if you're going to draw something that takes several strokes where you have to lift up and put the pencil back down, you need to be able to use vision to guide your actions. So I need to guide this from here to here. I lift it up, I need to guide it back here and then here and down. That's a lot of visual motor control. That has been damaged because RV damaged her dorsal stream. So I like to say that her drawing is basically similar to if I tried to draw something like this with my eyes closed. So could I do that? Yes. So my visual motor control is gone with my eyes closed. I'm now going to rely probably more on proprioception to guide my movements. And I can draw something, triangle-like, that's going to look like a boat, but I'm going to struggle to kind of move my pencil around and put it back in the right spot without vision. And that's why she's quite poor at these drawings that take, multiple shapes. The apple's okay because with your eyes closed, you can kind of do a circle, without visual motor control. And then putting the stem and the leaf on, well, that's a little bit hard because you can't tell exactly where you drew the apple. So poor visual motor control. Here's RV. We have pictures of her doing the mailbox task. And here we can see this is the condition where we ask you to mail the letter. And I don't know why, but they just asked her to mail her hand. Maybe they didn't run out of letters. So we have the mail slot, every trial, random orientation, and we can see she's not doing well at this task, right? She's hit the top, she's got the completely wrong orientation, so very poor visual motor control. Now we don't have pictures, but in a graph in a second we'll see that her perceptual estimate was great. So if she's not mailing her hand and we just say, what is the orientation of that mail slot? Boom, she would nail it. And then it's vertical, she'd say, oh, it's vertical. That was easy for her because that requires her intact ventral stream. So this is a summary of her data. And when we ask her, what is the orientation of the mail slot? She's great. And then we say, okay, well, go ahead and post your hand. And you would think, the opposite of DF is that if you know the orientation, I know it's horizontal, you should be able to use vision to guide your action towards that. But she can't. It's a bit of a paradox because knowing what the orientation is, well, that's her ventral stream guiding her visual abilities. But then actually posting the hand, even though the ventral stream knows, that's not going to help because the dorsal stream does that. And for her, the dorsal stream is damaged. back to the domino task, we'll call it, and we'll look at the two versions of the task. So estimate the size of the target. That requires the ventral stream. And then we'll go ahead and pick up the target, or the domino. So we looked at control before. This is an easy task. RV, what happens when we ask her to grasp the object? She needs a dorsal stream. Her dorsal stream is damaged. So she does not scale the size of her grip aperture to the size of the object. She just opens her hand big and hopes to touch it. Because if she touches it, she can then use her sense of touch. But if we ask her a perceptual estimate, okay, there's a domino in front of you. How big is it? Small domino, small grip aperture. Big domino, big grip aperture. So this is perfectly good scaling, comparable to control, showing that yes, her ventral stream is fine, but her dorsal stream is damaged. So that's RV, basically the opposite of DF. I think in a perfect world, they would live together, and one of them could do all the dorsal tasks, and one of them could do all the ventral tasks. So a little clue to help you remember DF and RV and which condition which one has comes by chance by their initials. So DF, what you can do is remember she has a D in her name, so that means her dorsal stream is intact. And perhaps it would be better if it told you what was damaged, but these are actually their initials, so we have to kind of use this memory technique. So if her dorsal stream is damaged, that means if the dorsal stream is intact, that means the ventral stream is damaged, and we call that visual form agnosia. RV, by chance, has a V in her name, so that can remind you her ventral stream is intact, her dorsal stream is damaged, and we call that optic ataxia. One more DF and RV experiment. And this one was important because they tested DF and RV on the same task in the same published study. Some of these other experiments, they had one study with DF doing the mailbox task versus control, and then later on they tested RV on the mailbox task versus control, but they were separate studies. This one, they both participated in the same study, in the same publication, which strengthens the results. So the first task, they had these two-dimensional shapes, kind of think of them as potato chips, and they would put two of them beside each other, and you had to decide, are they the same or are they different? So this is a perceptual estimate. You need to know, the shape of the object. So DF and RV, they can see these, but to know whether they're the same, you need your ventral stream. So let's use our clue here, our memory technique. RV, her ventral stream's intact, so she'll be good at this. DF, dorsal stream intact, damaged ventral stream, so she'll be bad at this. One other thing, this task, this is a very easy example here. These are obviously the same, these are obviously different. They made this task quite hard so that controls, someone with an intact dorsal and ventral stream, were also only 90% correct. So RV, this is actually comparable performance to control because the task was harder than just these two easy examples. So these results do show us, yes, DF is bad, 52%, she's basically guessing, and if you asked her confidence on any trial, we'd say, are they the same or different? She'd say, different. We say, how confident are you? She's like, I don't know, I'm guessing. So again, she knows that she's not good at this. All right, now we're going to test their dorsal stream. So what, kind of different from the other task, but here we're going to say, go ahead and pick up the object, but we haven't really scaled the size of the object. What we're going to do is look to see, does your grasp make sense? And let's find an example here. So let's go with this. Okay, so we have some calipers here. So if I'm going to pick this up, to make it easy and safe, I don't want to drop these expensive calipers, I want to pick it up so that the mass of the object is distributed, it's half on one side, half on the other. So what I'm probably not going to do is try to pick it up from here, right? Because that, technically I can do it, but it takes way more force. And this is maybe not the best example because so much of the weight is on one end. But let's go with, well, I guess we can go back to a pencil. So if you're going to pick up a pencil, you're probably going to pick it up in the middle so that your grip aperture bisects the center of mass. It's just a normal way to pick things up. Pencil's not a great example because it's so light, but okay, here we go. We got a Lego space shuttle. So if I want to pick this up, actually you can already see, I picked it up in the middle because I don't want to drop this, right? I'm not going to pick it up here. I'm not going to, I guess the back's not too bad, but you usually pick things up in the middle. So down here we have control participants picking up these kind of potato chip objects. And there's no one right way, but it's typical to put your fingers on both sides and bisect that mass in a way to make it easy to pick up. So if you look at all these, the lines, they often go through the middle of the object, and that makes sense. So this is good performance. Now DF, her performance was also good. So look at these lines bisecting, the center of mass. And these objects, the mass would be evenly distributed, unlike my caliper, so it's a little bit of an easier example. So why is she good at this? Well, to pick it up, you need your dorsal stream. DF has an intact dorsal stream. RV has a ventral stream, but not a dorsal stream, so she'll be bad at this task. And if we had video of her doing this, you'd see very poor visual motor coordination. And from these graphs, even though, like, say these ones look okay, some of the ones do go through the center, this one's way off, these ones are way off, that's way off, this is way off, so is that. So sometimes she gets it right by chance, but overall it's quite poor compared to control or even to DF. And if we had video of it, it would look way less natural and coordinated than DF and control. So this dissociation in DF and RV, we call it a double dissociation because what one lacks, the other has intact. So DF, visual form agnosia, intact dorsal stream, so she'll be good at visual motor control or these how tasks. And if she's good at this, she's got to be bad at perception, these what tasks. RV, who has optic ataxia, so her ventral stream is intact, so perceptual estimate will be good, and conversely, visual motor control or these how tasks will be bad. So if we kind of pull back to the overall visual schematic, so which areas are involved in processing vision? Vision starts at the retina here at site number 1, and then we're going to see this division between our cortical pathway and our subcortical pathway. So the subcortical pathway, and we're skipping some of the smaller structures, but we go to the lateral geniculate nucleus and then up to the pulvinar nucleus. Superior colliculus, got that wrong, to the pulvinar nucleus. Now the cortical pathway, that's the one that goes to the lateral geniculate nucleus and then to the primary visual pathway. And that was kind of as far as we got in our last module. And now we've seen that from the primary visual cortex, we have this dorsal stream that goes to the posterior parietal cortex, and we have a ventral stream that goes down to the inferotemporal cortex. So that's a lot of information about DF, RV, dorsal and ventral stream. So if you want, you can take a break now, look back on the material, study it a bit, and there's a section of practice questions you can use to test your knowledge on what we've been through so far. Now in the second half of this class, we'll eventually come back to DF and RV, but we're going to look at dissociations between dorsal and ventral functions in neurologically intact individuals. And this is interesting because we can show, in even you and I, that the dorsal and ventral streams will have differences of opinions on the same task or similar tasks. And we don't have to go to the extremes of damaging the dorsal or ventral stream to show that how we see these, how our mind perceives kind of how and what can be different. The way we see this is with visual illusions. And we'll look at two versions of this. The first here is called the Ebbinghaus illusion. And you've maybe seen this one before, it's nothing to write home about, but the question or the illusion is, if you're comparing these orange circles, which orange circle is larger? The one on the left or the one on the right? And you've maybe seen this before, so you might already know the trick to it, that the orange circles are identical in size. So I made this in PowerPoint, I copied and pasted that orange circle. Here's some horizontal lines to show, yes, they're the same size. But there is a bit of a perceptual illusion here. Because there are large objects around this one, it looks relatively smaller. And because there's small objects around this one, it looks relatively larger. So this is one version of a size contrast illusion called the Ebbinghaus illusion, and we'll look at a second example of this same size contrast illusion later. But let's look at how the dorsal and ventral stream think about this illusion. So we've got a pretty typical dorsal ventral experiment. In one condition, what we've done is imagine these are like poker chips, the middle circles. And in one condition, we'll say, oh, on this trial, pick up the one on the left. And you'll go and you'll reach and you'll pick it up. That's a how ability, it's a visual motor ability, so you'll use your dorsal stream to do that. In another condition, we'll just say, okay, the one on the left, for example, on this trial, other trials we might ask you the one on the right, how big is that poker chip? So don't go ahead and reach it, just make a perceptual estimate of its size. That requires the ventral stream, and you'll, even without thinking about it, you'll use your ventral stream to accomplish that task. All right, let's look at the results. So we did this with young adults, neurologically in task. I pretty much guarantee if I tested all of y'all at home, we would find these same findings. So first we say, okay, how big do you think that object is? And this is where we see the illusion. So there is an illusion that's going to affect your perception of these objects. So one condition, you open your hand bigger than the other. Now what are these conditions? So the white condition here is this one, so they're small circles, so remember it makes it look relatively larger, and look, you're making your hand, your grip aperture, a bit bigger. When the black condition, that's where you've got these big circles around, so it makes the center circle look relatively smaller, so you open your grip aperture a little bit smaller. So this is confirming, yes, there is a perceptual illusion, which hopefully you can kind of see in the slide itself. It's one of those illusions where once you know that they're the same size, it's not like the perceptual illusion disappears. Now what about if we ask you to go ahead and pick up that poker chip? The interesting thing is your maximum grip aperture is identical in both conditions. The visual illusion does not fool your grasp. So just to review this here, we have this size contrast illusion, here the Ebbinghaus illusion, and the ventral stream was fooled by the illusion. When we ask you, hey, how big are those middle things? Perception, the ventral stream, your what abilities are fooled. When we then say go pick them up, the dorsal stream takes over and your dorsal stream is not fooled. And this is where it's kind of fascinating, because if you think of your brain, as two different people, a dorsal and a ventral stream, the dorsal stream's like, hey, there's no illusion. The ventral stream says, yes, there is. You would think that those two individuals, those two parts of your brain, would communicate with each other and resolve their differences. It's kind of like if you're going to vote and half of your brain wanted to vote conservative and half your brain, Republican, let's get the right American terms here. Half your brain wanted to vote Republican and half your brain wanted to vote Democratic. There'd be some sort of internal battle, and eventually you decide who you want to vote for. But here the dorsal and the ventral streams have two opinions on what they see in front of them. And if that needs to be expressed in a perceptual estimate, then the being fooled by the illusion appears in your perceptual estimate. And then if we move to pick up the objects, the dorsal stream takes over, and all of a sudden we're not fooled by the illusion. So a weird contradiction. So these are pretty cool experiments because we can show this dissociation between perception and action even without having to rely on these very rare individuals like DF and RV. There's a huge body of literature and research on visual illusions because they kind of tell us about how our brain perceives reality. And the overall finding is that we don't see reality. Information from the world comes in, and then our brain tries to interpret what we see. And we're pretty good at this. And most of the time, we do perceive the truth, or we see reality. But there's lots of ways, like an Ebbinghaus illusion, which isn't that complicated. You know, hey, put some big things around something, or put some small things around it, and all of a sudden we've fooled our brain. So it's not actually too hard to fool the brain. And we see that with many examples of illusions. And they all kind of, besides dorsal ventral examples, show us that what we perceive is not necessarily reality. It's what our brain and our mind is interpreting based on the information given it. So just a couple illusions just for fun. So here, this illusion, if you look at one area of it and focus on it, the other areas look like they're moving. And then if you then look at an area that was moving, it stays still, but now the areas in peripheral vision, they look like they're moving. And the idea behind this illusion is that for some reason in peripheral vision, your cone cells, the ones that see the colors, tire out at different rates. And that's really emphasized here by having these, very contrasting colors. And when one type of cone cell tires out and can no longer see a color, but the other cone cells are still fine, our brain gets confused into thinking there's motion. Okay, let's put that away before I get dizzy. There's a website that does an Illusion of the Year contest, and in the description are links if you want to check out these videos. I won't test you on them, but this is from a Japanese researcher. He's won or placed highly in the competition a few times, and these are kind of two of my favorite entries by him. But you can also check out any year's competition or their overall top-rated illusions if you've got too much time on your hands. Right, back to using illusions to look at the function of the dorsal and ventral streams. And we're going to see there's one more distinction between what the dorsal stream does and what the ventral stream does. And here we're using a flanker illusion. It's a size contrast illusion, just like Ebbinghaus, but maybe less visually powerful. So here's the flanker illusion. So imagine we've got these two dominoes side by side, and you're always going to reach for the one on the right. But the one on the left, if we make it smaller, it's going to make this domino seem relatively larger. Kind of the same principle as the Ebbinghaus illusion. Now over here, this domino, it's actually the same size as this one, but if we flank it with something larger, it looks relatively smaller. And if we asked for your perceptual estimate here, we would see that you would say, yep, this one's a little bit bigger, this one's a little bit smaller, even though they're exactly the same size. And in this experiment, they didn't bother testing your perceptual estimate because it was well documented that yes, there is a visual illusion to your perception. They're going to focus on action and presumably the dorsal stream. So here's the task. You've got these two dominoes in front of you. Again, you're always reaching for the one on the right. The flanker is to either present an illusion that this would be smaller or larger depending on the size of the flanker. So there's going to be two conditions, and both of those you're going to go out and pick up the object. And the idea is that, well, you're going to use your dorsal stream. That's what they suspected. Because you use vision to guide your action, that's the dorsal stream. But what they're going to do, different than other experiments, is you either will have vision or not have vision as you prepare your movement. And that's shown in these schematics, but let me show you in PowerPoint first, and we'll come back to what this schematic means. So here's the vision condition. So you're starting with your thumb and index finger together at a starting position. You know you're going to reach for this one on the right. You get a go signal. You then prepare your movement, and then when you're ready, you start to move. So you have vision during your reaction time. And then as soon as you start to lift your hand, you lift off a button, and that button triggers these spectacles to close, and you'll have to do the rest of your reach without vision. So they don't turn the lights off in the room, so it's not as extreme as this. But just imagine you see the object, you prepare your movement, you start to execute that movement, boom, and then the glasses close, and you have to reach for the object without visual information. In the occlusion condition, the critical difference here is you lose vision earlier. So you can see the objects, you have to move to the one on the right. You get the go signal, and as soon as you get the go signal, the spectacles close. You now have to prepare your movement without real-time visual information. You're going to use your memory, and then you have to execute your movement without real-time visual information. So that's depicted in these schematics here. So when this box is up, that means you have vision, and when it drops, that means you've lost vision. So the key difference is, do you have vision when you prepare the movement or not? In the vision condition, you do. In the occlusion condition, you do not have real-time visual information as you prepare that action. Their results, they did a touch of math. We need to cover this, but thankfully it's just a subtraction. What they do is they take your grip aperture in this condition and subtract it from this condition. Now let's say there's no illusion on your movement, and that's what we would predict when you use your dorsal stream. So your grip aperture, let's say here it's 5 centimeters. Over here it's also 5 centimeters because these dominoes are the same size. So we subtract 5 from 5, we get this difference score of 0. That suggests there's no illusion. Now if there is an illusion in your grasping movement, then you would probably think this one looks a little bit bigger because it has a smaller flanker. And this one looks a little bit smaller because it has a bigger flanker. So maybe your grip aperture here is 6 centimeters, and over here it's 4 centimeters. So we subtract these two conditions. We've got 6 minus 4, we get 2 centimeters. So if we get a score significantly larger than 0, that suggests your hand was fooled by the illusion. Just to summarize these hypotheses here, so both conditions you're reaching, and the idea is the investigators thought, because you're doing a visual motor control, you'd use your dorsal stream, as we've seen before, so you should not be fooled by the illusion. However, if in one of the or both these conditions we see that you are fooled by the illusion, that must mean you used the ventral stream to move, because remember, your ventral stream is fooled by illusions. So here are the results, just two bars. So here, this is the condition where you have real-time visual information as you prepare your movement, and this was comparable to 0. So they said, okay, different score of essentially 0, no visual illusion, that must mean you used your dorsal stream. And that kind of makes sense with everything we saw with DF and RV so far. But check out this other condition. This is where you have no real-time visual information as you prepare your movement, and we see a score significantly larger than 0. So they said, okay, when there's no vision during movement preparation, you are fooled by the illusion. And it's strange because if you're fooled by the illusion, that means you must have used your ventral stream. And all of a sudden you're using your ventral stream to make a guided movement, which was odd. They thought all movements would be controlled by the dorsal stream, but that's not the case. So here's a good quote from the article by Goodale and Westwood. So visual motor networks in the dorsal stream operate in real time. These networks appear not to be engaged unless the target object is visible at the exact moment the response is required. In other situations, for example, memory-driven, which when those glasses close, you have to use your memory to prepare your movement, the control of action passes to other systems that access a representation of the target object laid down by the perceptual mechanisms of the ventral stream. So maybe a little wordy, but it's nicely explaining these results. If you have vision when you prepare a movement, you use your dorsal stream, and then there's no illusion. But if you have to make a movement based on a memory of where you saw the object, so the glass is closed, oh, I remember it's there, so I'm going to use my memory. Well, all of a sudden, your mind decides, I'm going to use my ventral stream for that memory, and now you are fooled by the illusion. So if we put this into kind of our typical schematic here, we have a distinction here. So it's not that all action is done by the dorsal stream, and that's kind of what we said when we talked about DF and RV. But through further research, we've seen that, well, most action is done by the dorsal stream. And we call this real-time action. So when you make a movement and you have real-time visual information. And this is typically how we move. You know, if you think of an athlete playing some sort of ball sport, let's say tennis, you know, you're not going to close your eyes and chase down the ball. You know, that's just not going to work out very well. But we do sometimes make memory-guided actions. So let's say you walk into a dark room and you're just using your memory of where the light switch is, and you move over to flick it on. Or maybe in the middle of the night your alarm clock goes off, or your alarm clock on your cell phone goes off. You know, there's no light or anything, but you have to move to remember where you put your phone. So you are making these movements. You know, you would think it's this how ability, and it kind of is, but because it's memory guided, you're going to use your ventral stream to do that. So yes, most action is done by the dorsal stream, or more specifically real-time action. Memory guided action is actually handled by the ventral stream. Let's take this back to RV, and this is actually good news for RV. So RV, so she has a V in her name, so her ventral stream's intact, her dorsal stream is damaged, and previously saw that her visual motor control was poor. The good news is that her memory-guided action is preserved, and she kind of figured this out for herself. So if, let's say, we say, okay, RV, here's a pencil, you know, try to grasp it. If she looks at it and tries to grasp it with real-time visual information, she's trying to use her broken dorsal stream, and it's going to be, you know, poorly coordinated. You know, she might even miss it. It's not good. What she can do, because she knows her memory-guided action is fine, you can say, okay, you want me to pick up that pencil? She can close her eyes, and now she'll guide her hand there with her memory. Now, memory-guided action is obviously, where is it? Okay, there it is. Not as accurate as real-time visually guided action if your dorsal stream works. But for RV, memory-guided action is going to be better than real-time action because her dorsal stream is broken. Now, if she does use this, she doesn't typically close her eyes to move to something because that's a bit extreme. All of a sudden, you know, you don't know what's happening in the world. So what she'll do is she'll look at the object she wants to reach to, look away, so she can still see the world. She doesn't have to close her eyes, but because it's outside of her real-time visual field, she's using her memory to go pick up that object. So this is good news for RV. So let's confirm those results with RV. So we could have potentially tested her on the exact same experiment. I'm not sure if they have, but we have another experiment that did kind of the same thing. So in this case, RV was reaching for objects that could be small, you know, medium, or large. So there was a range of sizes. And we wanted to see, did she scale her grip aperture? So we do a classic dorsal task. We say, okay, look at the object and pick it up. She's bad at this because her dorsal stream is damaged. We have poor scaling of her grip aperture. It's kind of all over the place. Like, look at this trowel. So it should be a medium object. This trowel, she thought it was extra large, and this trowel, she thought it was medium. Not good. But then we say, okay, look at the object, close your eyes, and pick it up. And now we see pretty good scaling of her grip aperture. When it's small, she makes a small grip aperture. When it's large, she makes a large grip aperture. This wouldn't be as good as a control person doing real-time action because, you know, closing your eyes and reaching for something is not as good as opening your eyes and reaching for something for a control individual. But we do have confirmation here that yes, she's good at this task, as good as a control person reaching, you know, for something with their eyes closed, because she's using her ventral stream, and of course, RV has an intact ventral stream. Okay, last summary for DF and RV. So if we look at their perception, you need a ventral stream. So RV will be good at it, DF will be bad at it. Okay, next thing we can ask them to do is make a real-time action. So they have their eyes open, they're looking at the mailbox. So this will require your dorsal stream. DF has a dorsal stream, she'll be good at it. RV will be bad at it. And then finally, memory-guided action. So not the most common sort of movements we make, but it shows us something interesting about the brain. So to do these, remember you need your ventral stream. RV has a ventral stream, so she'll be good at it. DF will be bad at it. And the way they're trying to demonstrate that it's memory here, because you can't really tell whether the eyes are open or not, but see the mailbox, how it's dotted here? They're trying to say she saw the mailbox, closed her eyes, and then tried to mail the letter. Finally, I just want to bring up some criticisms of the ventral streams hypothesis, the idea that we have a dorsal stream, a ventral stream, that divide and conquer kind of our visual abilities. So for visual illusions, some people have suggested it may be unfair to compare perception to action because they're not identical. You know, they're apples to oranges. They're both fruit, they're both round, but they're not supposed to be the same thing. In cognitive psychology, you know, they have difference in task demands, attention, so it's not the most fair comparison. Now DF and RV are a great source of evidence, but they're rare individuals. There's been a few more people like them, but it's rare to damage all of the dorsal stream or all of the ventral stream and not other areas. And of course, it's not ethical to go in and create the exact damage that we would like to test. And one thing that's been noted with DF is when she's been retested years later, her dissociation between perception and action hasn't been as strong. Now, is that because her brain, some areas have recovered? Is it because her neuroanatomy has perhaps changed a little bit? Some connections have reformed around the damage? Has she discovered, you know, very subtle unconscious strategies that help her with the task? It's very hard to say, and it's also, again, hard to rely on people like DF and RV that are so rare. The other is that in these tasks, in these tasks, we do show this strong dissociation between perception and action, or dorsal and ventral, where these two streams will have differences of opinions and they don't resolve their differences. In other tasks, we can actually see communication between the dorsal and ventral stream. So they're largely independent, but remember, any area of the brain is going to be connected to any other area of the brain. The dorsal and ventral stream, you know, it's not like those two areas have a single neuron between them that will resolve differences. You know, they're more independent than that. But eventually, through enough connections, you know, information from one would get to the other. So the tasks we've seen here, they do look very independent, but some other tasks can show them interacting. Just wanted to bring up those criticisms. Dorsal and ventral research is still alive and well. It's kind of a new generation of it, but we won't have time to discuss it in this course. All right, to test your understanding on kind of the second half of this presentation, take a look at the second set of practice questions, and good luck with the quiz.